It's a pleasure to be with you here this morning, and welcome alumni and guests. I want to congratulate whoever made this beautiful artwork, by the way. This is really spectacular. Let's see if we can get started. So I'm going to really try over the next 30 minutes or so to get you excited about the remarkable progress that we've made in cancer and the potential for treating cancer in the future. So I thought we would start with a few questions. How big a problem is cancer? What is cancer? Where does it come from? Why do we get it? And what can we do about it? And as Val alluded to, cancer has been with us for a long time. This is an example of a mummy from ancient Nubia from 1200 BC that shows metastatic lesions in the hip bone of and the femur of a deceased person, and you can see those lytic lesions that are spread throughout the bone. And even going much further back than that, there was a recent report of an osteosarcoma in a fossil from a turtle that is thought to be osteosarcoma from 240 million years ago. So it seems that cancer has always been with us as metazoans as we became multicellular and have all these complex interactions among our cells, that the risk of some cells becoming abnormal and escaping growth control has been a major problem. So what is the scope of the cancer problem in the United States? Well, in 2020, we had 1.8 million new cancer cases and about 600,000 estimated deaths, unfortunately. But the good news is that the peak cancer incidence and the peak mortality occurred in the 1990s and has been diminishing ever since. And in fact, since 2001, or between 2001 and 2020, cancer mortality has been reduced by 27%. That is a pretty remarkable accomplishment over that period. And there are multiple factors that can account for why cancer mortality has diminished during that period. One is diminished tobacco use. That's a principal driver, and that's due to educational campaigns. Another is increased screening with mammography and colonoscopy, for example, as well as vaccines for HPV and for hepatitis B virus, for example. And of course, new cancer therapies and treatments are also driving this reduction in mortality. And I should say that a lot of this progress is fueled by the remarkable investment that's been made by the National Cancer Institute over the past decades, and really an extraordinary amount of information that we've gained on cancer. Now, this reduction in cancer death rates can be seen also by gender, where you can see the green line represents mortality due to cancer for males, and the orange line is death due to death in females, with blue representing the total death. And you see this inexorable decline in cancer mortality. We also know that cancer is intimately linked to aging, and that's shown here with the age-specific cancer death rates by year. And of course, we're very protected against cancer during the first 4 or 5 decades of our lives, and then cancer becomes a much bigger problem as we age subsequently, and we can talk about why that is. Now, this is all the good news about how cancer mortality and cancer incidence is diminishing. But the flip side of this is that cancer cases are on the rise. And the simple explanation for that is due to the enlarging population in the United States and other countries, as well as the aging and graying of the population. So even though we're making great progress, we're seeing increased cases. So 1.8 million cases by 2020, a predicted 2.3 million cases in 2030, and 2.9 million by 2050. And in fact, we're expecting 21 million cases worldwide in cancer by 2030. Now, just to take a quick look at what types of cancers are the most abundant. You can see these are the top 6. So lung cancer is number 1, and this is shown for males and females. Colorectal cancer, number 2. Pancreas cancer, number 3. Breast cancer, number 4. Prostate, number 5. And liver and bile duct cancer, number 6. Now, what's interesting about this is that this is by death rates. This does not exactly correlate with the incidence of these cancers, and the biggest outlier here is pancreas cancer, which is thankfully still quite rare, but it becomes a major player in cancer mortality because of the very poor treatment options for pancreas cancer and the fact that it presents so late. Now, there are also important disparities in cancer mortality and incidence, and I'll just touch on a couple of those here. So for example, in terms of socioeconomic status, there's a 3x increase in mortality among men with less education. So socioeconomic status makes a big difference in terms of cancer incidence and cancer mortality. And there are racial and ethnic disparities. And for example, African American women show a 14% increase, and African American men show a 24% increase in cancer mortality. And so these are important issues that we are focused on in the Cancer Institute. Okay, now let's talk about what cancer is. And I know many of you are very educated in this area, but I thought just in terms of level setting, cancer comes from abnormal growths or tumors. And those tumors can become, frankly, malignant and to develop into cancer. And the best understood progression model for that is in colorectal cancer, where one has polyps that are benign, and those are the ones that we try to remove through colonoscopy. And those can become gradually more advanced and become dysplastic, frankly, malignant, and develop into adenocarcinoma, and then become more invasive and metastatic as these tumors progress. And this just highlights the importance of catching cancer early. Cancer cells have a very disorganized appearance under the microscope. So on the top left, you can see these normal, benign glands in the colon. You can see that the nuclei are arranged in this picket fence type way. But on the bottom, you can see colorectal cancer, where now there's a disorganized appearance of the cancer cells. The nuclei are no longer arranged in that structured way. They're larger and atypical, as we say. And then in the middle view, you can see that these are now invading muscle layers. We know that cancer is a genetic disease, and this is one of the breakthroughs over the past 3 decades. And it's characterized by abnormal chromosomes and mutant genes. So this is a karyotype assay called spectral karyotyping, where each chromosome in a metaphase spread can be painted a unique color. So it's a very pretty picture. And on the left is a normal karyotype with 23 pairs of chromosomes. On the right, you see a characteristic karyotype from a patient with cancer. And there are many features here that are characteristic of cancer. One is an abnormal number of chromosomes. So you can see for chromosome 1 now there are 4 chromosomes. Chromosome 2, there are 3 chromosomes. But in addition, you can see that the chromosomes themselves are striped in many cases. And that means that they are now comprised of multiple different chromosomal segments that they should not be arranged that way. And those are called non-reciprocal translocations. And if one were able to look now at a more refined level, you would see that there are amplifications of gene regions and deletions of gene regions. And that amplification, gain of information, and loss of information in the sense of deletions is very, very important in driving cancer. Now, a very specific type of translocation is called a balanced translocation or a reciprocal translocation. And this occurs in many cancer types, in pediatric cancer, in some sarcomas, in hematopoietic cancers. And the first and most classical one is called the Philadelphia chromosome. And this is a reciprocal translocation between chromosomes 9 and 22, where information is now exchanged to create these new chimeric chromosomes. And at that junction, the point at which those chromosomes have been joined, there's a new bit of information, a new protein called BCR-ABL, where 2 genes have been inappropriately fused together. And that's a key oncogene that drives a disease called chronic myelogenous leukemia. And I'll tell you a little bit more about that later in the talk. So our knowledge of cancer has really been advanced by our ability to sequence cancer genomes. And as you all know, the human genome is comprised of a repeating or a long sequence of 4 nucleotide residues, GATC. And there are about 3 billion letters in the human genome. Our ability to sequence genomes has accelerated so rapidly, and the cost has dropped so precipitously, as shown here, that it's accelerated at a faster rate than the Moore's law that's been made famous here in Silicon Valley for an acceleration of computing power. And so now we've been able to sequence thousands and thousands of human cancers, and really we have a roadmap of all the genes that are altered in human cancer. And what we know is that most cancers harbor thousands of mutations. So this really shows how dysregulated the process of cancer development is, how abnormal cancers are, and it also gives you an insight into the challenge in trying to treat this set of diseases once the cells have become riddled with tumors and so abnormal. Now, a lot of this basic knowledge has translated into an incredible array of new therapies. And here, I'm just trying to highlight many of the breakthroughs that have happened over the last 25 years. And I'm not going to go through all of these, but there are new therapies for cancer, including antibodies that we usually think of in the context of fighting infection. But those have been weaponized to treat cancer in terms of Rituxan and Herceptin. There are many new small molecules that have been developed to treat cancer, and I'll tell you a little bit about that. New immunotherapies, we'll touch on that. New CAR-T approaches to treat cancer. And this has accelerated so much that currently the FDA approves a new cancer drug about every week. And this gives you a flavor for how challenging it is to be an oncologist these days. This is why oncology has moved to specialization and subspecialization, because to truly understand how to treat and how, cancer patients and to use all of these new therapies requires a real sense of focus. So now I want to tell you 3 vignettes about new therapies in cancer to try to make you excited about the remarkable progress. And one is in what we call molecularly targeted therapy. And the take-home message here is that we are elucidating the fundamental causes of cancer. We understand the proteins that are mutated, that are driving cancer, and we're developing drugs that specifically target those bad actor proteins. So this is in part the work of Nathaniel Gray, a brilliant chemist whom I recruited from Harvard last year, and he's leading our efforts in the Cancer Institute to develop new small molecules that target cancer. So just to start, our general notion of how we treat cancer really is based on antiproliferative chemotherapy. And the concept here is that cancer cells are dividing rapidly, and so over the decades we've developed drugs that are rather indiscriminate in their ability to kill rapidly dividing cells. And these could be DNA damaging agents or agents that destabilize microtubules. Now, it should not be underestimated that these drugs have saved millions of people worldwide since they've been in use, but they induce collateral damage in healthy tissue, side effects. They have insufficient efficacy, and they are characterized by drug resistance. So they typically work for a time, and then the tumor becomes resistant to them. So an alternative approach is to use this smart drug concept, and the most famous and initial one is a drug called Gleevec, which targets this BCR-ABL kinase that I told you is encoded by the Philadelphia chromosome. So this small molecule binds to the active site of this enzyme in a lock and key way, and it has transformed the treatment of chronic myelogenous leukemia, which used to be a very difficult to treat disease, and now most patients are able to be treated in a way that their disease is suppressed for many, many years. It's also been used to treat GIST, sarcoma, and there are other ones that are, and these drugs are being used in other cancer contexts as well. So this is really an example of this class of cancer targets called kinases, which are key for intracellular signaling. They bind ATP as well as a substrate molecule. They transfer a phosphate group to that substrate molecule. And so the idea is to inhibit this process. So typically these are transforming cancer cells by acting through a signaling cascade. They bind a ligand and then they signal to key cancer proteins like RAS, for example, and they engage all of these downstream kinase and molecules and transduce that information down to the nucleus and to transcription factors. An activating mutation in one of these kinases will typically be driving cancer, and that can happen, for example, in EGFR in lung cancer as a prototypical example. So there are many, many classes of these protein kinases, and they've been mined as drug targets. There are many that have made it to market, not only for cancer, but for other diseases. Rapamycin for organ transplant, Gleevec, I mentioned. Dasatinib is a second generation for Gleevec, for CML. Iressa for lung cancer, Tarceva for lung cancer. These are EGFR inhibitors. So they've had a remarkable track record. In some cases, these drugs have phenomenal responses in the clinic. So you can have a patient come in with metastatic lung cancer or metastatic melanoma, get treated with one of these targeted agents, and the cancer just melts away. And it's really a remarkable thing to see in the clinic. In many cases, especially with solid tumors, that cancer will come back months later as the tumor has learned to become resistant. And then we are working actively to try to develop mechanisms to deal with that resistance. Now, there's another new technology that I just want to touch on to introduce to you, because I've talked about this lock and key approach, right? If you have an enzyme that has an active site, you can make an inhibitor that docks in that active site. But many times we're trying to treat a protein that is not in that class. It's not an enzyme. It doesn't have an active site that can be targeted. And so a new technology that's emerged is to degrade protein targets rather than to just inhibit them in this lock and key means. And so this can be called drugging the undruggable. And it comes from the story of thalidomide, which you may remember was a drug that was a sedative that was given to pregnant women in the 1950s and led to these terrible limb defects in the offspring of those women. It was later reborn as a cancer drug for multiple myeloma, and it turns out that the way that this works is that it's a small molecule that's linked to cellular machinery that's degrading proteins naturally, and it can actually connect this degradation machinery to a particular set of protein targets. So that's how that drug is working. So then this whole scheme now has been exploited in such a way that one can develop new small molecules linked to a thalidomide-like compound that would then bridge the degrading machinery to a new protein target of interest. For example, a cancer drug this is also applicable beyond cancer, but essentially doesn't just inhibit the protein of interest, but it degrades it. It makes it go away. And this is a really exciting technology that Nathaniel and others are pioneering. So now I want to end by talking about immuno-oncology, immunotherapy, and how we have been able to harness the immune system to fight cancer. It's really a remarkable story. The history of immunology and cancer goes back 100 years, many, many fits and starts in terms of high hopefulness that the immune system could be exploited to cure cancer, and then abject defeat when that didn't turn out to be the case. But fortunately, many scientists soldiered on, and I want to tell you first about this remarkable set of discoveries and breakthroughs called immune checkpoints. This is a way of releasing a break between the T cell and the tumor cell. So we know that there are T cells, classical infection fighting cells, that are recognizing the tumor in many cases, but are not able to really be activated to kill tumor cells. And that's in part because of these negative signals shown here. One is CTLA4, one is PD1 and PDL1, that are blocking the ability of the T cell to respond to the tumor. So Jim Allison and colleagues found a way to make an antibody to these targets, again, using an antibody as a drug to block this interaction, to relieve this negative regulation between the T cell and the tumor cell. And when those antibodies are given to take this break off, this really enables the T cell to become activated to kill the cancer cell. And this has really been a remarkable revolution, and it came first with the observation that antibodies to CTLA4 could treat patients with metastatic melanoma. Metastatic melanoma was a very, very difficult disease to treat. It wasn't really responsive to chemotherapy. And this was helpful in about 10 to 20% of patients. And then it was found that another checkpoint, anti-PD-1, also could do this in melanoma and had even broader activities in some other cancer types, lung cancer, bladder cancer, kidney cancer, et cetera. And finally, the combination of these two, anti-CTLA-4 and anti-PD-1, showed even greater responses. And some of that primary data is shown here from a New England Journal paper, where you can see that the activities of antibodies to CTLA-4 and antibodies to PD-1 are additive or synergistic in leading to about 30 to 40% long-term cure in these patients with metastatic melanoma. And this is a remarkable change in how we view this disease. One of the best known patients was Jimmy Carter, who had metastases to the brain with melanoma. That was years ago. He was treated, and he's been in long-term remission. And there are many, many other such stories as that. Now, the flip side is that about 60% of patients are not effectively treated with this approach, 60% of melanoma patients. And so it turns out that responsiveness to checkpoint blockade varies widely. Many tumors are not responsive to this. Many different cancer types are not responsive. And it turns out that tumors that are responsive have a very high mutational burden. So in this case, even though I told you earlier that it's bad that cancers have thousands of mutations, in terms of responsiveness to checkpoint inhibitors, that's actually a good thing because it leads to the expression of new epitopes that the immune system can respond. So these cancers that have a lower mutational burden are typically the ones that don't respond, whereas those that have the high mutational burden are the ones that do respond. Now, another story that's related to this is that it was found that cancers that have what are called mismatch repair deficiencies, that's a defect in DNA repair, that actually is good for the tumor because it creates a lot of mutations that can drive cancer gene function. But here it's creating new epitopes. And so when you have this very high mutational burden from mismatch repair deficiency, that really leads to high response rates. And so that was a very exciting discovery a few years ago. And this is just a slide showing what this mismatch repair process does in terms of repairing DNA. Okay, and then within melanoma patients, I told you that some patients respond and some don't, and it's shown that patients who have high number of mutations are the ones that typically respond better, and those that have a low mutational burden are typically the ones that respond worse. And this is true in patients with non-small lung cancer as well. So really, really exciting progress here in just simply giving an antibody to a patient to activate the patient's own immune system. That immune system is primed to respond to the tumor, but unable to do so effectively. And that's probably because the immune system, the cancer rather, has learned to become resistant to the immune system and to inhibit it. A different immuno-oncology approach is one called CAR T therapy. And this is one that is being pioneered by Crystal Makel, whom we recruited from the NCI to the Stanford Cancer Institute about 5 years ago. And Crystal has built a remarkably successful and exciting program here at Stanford for children and for adults using CAR T therapy to treat cancer. And this is a different concept. It's really cellular engineering. It's a way of taking a patient's immune cells, engineering them in the laboratory to fight cancer, and then reinfusing those cells back into the patient. And here, I'm not going to go into too much detail, but on the left is the natural T-cell receptor response, how it engages with what's called an antigen presenting cell, and it does so using the T-cell receptor that recognizes a molecule called MHC, and then a peptide, which is an antigen that's expressed in the antigen presenting cell. So the synthetic CAR approach is different in that it doesn't rely on the natural T-cell receptor. It uses something called a chimeric antigen receptor, shown where it says co-stim1, And it has its own binding specificity that's targeted not against MHC or a peptide, but against an antigen on the surface of the cancer that's typically expressed in the whole lineage of cells. So for example, if the tumor cell is a B-cell lymphoma, then this CAR is directed against CD19, which is an antigen expressed on all B cells. So it's a way of training T cells now to go kill B cells in this case. And this process is done here on campus in the laboratory. So a patient will have his or her blood drawn. T cells are isolated. They're stimulated in culture. They are then transduced with a virus that's expressing this CAR, this chimeric antigen receptor. Those cells, those re-educated cells are then expanded in the laboratory and infused back in the patient where they home to the tumor and kill the tumor. And this has been really a remarkable set of activities and led to many, many approvals, principally for lymphoma and for some leukemias and for multiple myeloma. Now, we would like to use this to treat all cancer types, right? But so far, the extension of this exciting technology to solid tumors, solid cancers, breast cancer, lung cancer, pancreas cancer, et cetera, has been very negative. So there haven't been any real responses yet, despite a lot of effort. But I would say that this effort is just getting started, and there's still a lot of excitement that we can learn how to adapt this CAR T therapy to treat common solid malignancies. And just to give you a flavor of what's going on here at Stanford, this is a trial that Crystal and Michelle Monge are running using what's called a GD2 CAR. That's the name of the CAR. And it's being used to treat a remarkably challenging disease. It's a pediatric brain tumor called DIPG. This disease typically occurs in young children or teenagers or young adults. And here's an example in the brainstem of a tumor. Before this patient was infused with these lymphocytes that have been re-educated to fight the tumor. And then you can see at 3 months and 4 months how this patient has improved radiographically and also clinically in terms of an improvement in hearing, facial sensation, chewing, speech articulation, walking, et cetera. So this is an example of how we can take really exciting science at Stanford, translate it, develop clinical trials, and then test those in our patient population. So I'm going to summarize what I told you, which is that an incredible investment and progress in understanding cancer is paying off. It's translated into improved survival for cancer, to the development of new targeted therapies, new immunotherapies. It's making a real difference in the lives of cancer patients, and really there's not a more exciting time in the history of cancer research or cancer therapy to be working in this field right now. And I want to thank you for your attention, and I'm very happy to take any questions. I think we have a mic. We have mics around the room, so please don't be shy. Just raise your hand. You'll get a mic, and you can shout out a question to me.
If people aren't dying of cancer and heart disease, what are they dying of? I thought they sort of had to pass away from.
Yes. Well, they are certainly dying from COVID, unfortunately. That's been the uptick in mortality over the past couple of years. But what are the other major causes of death? Well, it's going to be certainly infection, pneumonias, other chronic conditions, neurodegenerative disease. One could imagine that if we can fix cancer and heart disease, that neurodegenerative disease might emerge as a large cause of death.
You brought up CAR T cells there, and
Yes.
also T cell energy and penetration of solid tumors.
Yes.
There has been a lot of work recently in scientific laboratories reported in Nature and Science. We're making some progress on this. What is your opinion, and how soon do you think this will occur in a really clinical setting where we can knock out solid tumors?
Good question. So the question, I'll just repeat it, is about the progress and exciting advances in T-cell therapy, and when do we think that this will be broadly applicable to solid tumors? It's hard to make predictions because they sometimes are awaiting a transformational breakthrough, and then all of a sudden things progress very, very rapidly. I think the challenge on the solid tumor front is that one could be penetration, as you say, that the T cells are not penetrating the tumor effectively. That might be more difficult to overcome. The other challenge is, what is the antigen target that one can use? So I showed you that most of the progress has been made in B-cell lymphoma. You can target B cells. We can live without B cells, so we can infuse CAR T cells that will eliminate all our B cells, and we can survive with that partial immunodeficiency. Solid tumors are more difficult because you have to find an antigen that is somewhat selective to the cancer. For example, you wouldn't want to treat with a CAR that could kill all your liver cells if you had liver cancer, because you wouldn't want it. You can't live without a liver. So that's the challenge in finding this therapeutic index for a CAR and tuning it to be responsive against the cancer without really being reactive against the entire solid organ. So I think that there are a lot of advances in tuning CARs that may pay off in that regard.
Great. Thank you for an outstanding talk. Just a quick question. Which cancers and which technologies do you think would be most benefit from CRISPR technology?
That is something I'm actively thinking about. CRISPR is being used in CAR T therapy right now. So I quickly went through the scheme of how we make CARs, and right now it uses lentiviruses, right? That's always risky because you're doing random insertion of a virus into a genome. Could disrupt or activate genes that would lead to cancer in the CAR Ts themselves. We haven't seen that, but we've seen that in other gene therapy contexts for sure. The next generation will be to use CRISPR to modify those CAR T cells in a safer way. So that is definitely coming. But I think that CRISPR could have broader uses as a cancer-fighting tool, and those remain to be determined. But it's an interesting concept.
Thank you again very much for the talk. I was wondering if you can go a little bit more into what Stanford is doing through the Cancer Institute.
Oh, yes. Thank you. Be very happy to. So the Cancer Institute is the organization on campus that really structures and organizes everything from clinical care in cancer, to basic science in cancer, translation, clinical trials. We have about 300 faculty members who are members of the Cancer Institute. We have about 400 people who actually work in the Cancer Institute in the organization. We run the whole clinical trials infrastructure. So if you know someone who's had a clinical trial, that's been really done through the SCI, the Cancer Institute. We have about $200 million in funding from grant agencies annually that support our cancer research. And we give out grants. We give out seed grants, innovation grants to help catalyze new discoveries, to bring new teams of people together. We try to bring scientists and clinicians together to develop new ideas. We help support trainees, fellows, for example, who need bridge funding during their training periods as they're trying to make it to a faculty position. And we are what we call the NCI Comprehensive Cancer Center. That is the highest designation from the National Cancer Institute to about 50 cancer centers nationally. And that's what I did when I started about 3 years ago. My job was to renew our comprehensive status, which we just successfully did. It requires a big team approach to do that, and it's recognition of the outstanding clinical care and cancer science and clinical trials that we have at Stanford. So thanks for that question. Yes. Oh.
Yeah, it seems like it's kind of the wild, wild west right now for precision diagnostics for cancer. I wonder if you could just kind of give just a real brief overview. You got cell-free DNA, the mRNA, microRNA, exosomes, all these precision diagnostic tests for cancer, where you sort of think the horse race is at this point.
Well, that's a really good question. Some of those technologies that you alluded to are more research-based, not really used for clinical care right now. What we're doing at Stanford is that we have developed our own designer panels for high-throughput sequencing, and we sequence patients' tumors on this panel. It's called STAMP, and it's run out of our pathology labs in a CLIA-specific way. And that interrogates a panel of about 250 known cancer genes, and that information then is translated to the doctor so that the doctor can make specific decisions about which one of those therapies, those targeted therapies, to use. So that's the state of the art in terms of what we're doing. There are much deeper ways of interrogating cancer, and we do that in a research capacity, whether it's whole genome sequencing or technologies called ATAC-seq that was developed here at Stanford, which really probes the chromatin patterns, chromatin accessibility patterns throughout the genome. There are ways of doing RNA-seq, single cell analysis, so now we can sequence the RNA in a single cell, which seemed crazy about 10 years ago, but now it's common. So there are many, many ways of getting incredibly deep information about cancer. Not all of those have translated clinically yet, so we don't do those types of very in-depth, expensive approaches to every cancer patient. But we are right now actively thinking about what does the future look like in terms of which ones of these technologies could provide information that would be clinically actionable and help the patient and help the doctor in clinical decision making. So each of these approaches leads to just a tsunami of data, and then at Stanford we're really experts in the biomedical data sciences. We're trying to match up that capacity with our genomic sequencing capacity so we can bring the best minds together to solve these very complex problems. Yes.
The last 30, 40 years, the PSA for prostate, I don't know if that's what caused the prostate to go from number 2 and going down to 5. Is there any other, well, how's that doing? Are there any other markers of any other tumors?
Yes, so the question was about prostate cancer and PSA. PSA is a remarkably good blood test at finding prostate cancer, and that's, if you look back at the incidence of prostate cancer, it spiked with the introduction of PSA because now we could find all these men who have prostate cancer. The complexity there, I think, is characteristic of the complexity in detecting cancer earlier and earlier. Because if you can imagine finding cancer early, it's very useful because we can cure patients, but it also can lead to significant overtreatment because many of those prostate cancers won't actually be an issue medically for the man in question, right? And that's what's led to more of a watch and wait approach to certain prostate cancers when they have more, less aggressive features. So it's a very, very good test at finding prostate cancer. It's helping some men. It's leading to overtreatment. There's a lot of work going on to try to develop new technologies to try to find cancer early. The best is probably the one issue that you raised in your question, which is on cell-free DNA. So now we have very powerful ways of isolating DNA that's been shed by tumor cells into the blood, deeply sequencing that DNA, and then trying to figure out, are there cancer mutations in that DNA? If so, what tissue does that DNA come from? That's probably going to be a very powerful technology to find cancer earlier and earlier. But I think it's going to lead to a lot of secondary and tertiary issues with what to do next with that information in terms of how to find the cancer. Once you find this early cancer, should you treat the cancer? Should you watch it? It's going to be a complicated world, I think, moving forward. Yes.
It's about your research on telomerase?
Yes, I'd be happy to. Thank you. So I'm studying an enzyme called telomerase, which is an enzyme that is adding repeats to the ends of chromosomes. It turns out that stem cells, long-lived cells in our bodies that need to fuel tissue renewal, need to express this enzyme because otherwise, without this enzyme, the actual tips of our chromosomes shorten a bit with each cell division, eventually leading to a process called senescence, and then those stem cells can no longer be functional. Cancer learns to upregulate that enzyme in almost every cancer, 95% of cancers. And they live off this ability to maintain telomeres and become immortal. So it's the key enzyme that leads a normal cell, to become immortal. And we're very interested in making inhibitors to that enzyme, which I think would be broadly useful to a number of human cancers. Thank you.
Another question.
Yes.
A lot of people are asking, when we have healthy people, California, we don't have many smokers, so there's always a challenge of looking for smoking cessation programs here. Still can benefit. But a lot of people do wonder about how can they be healthier and avoid these problems. And things like concepts that are very biochemical, molecular autophagy, and it's been a big topic recently outside of the medical realm of intermittent fasting. And I'm curious if you yourself or the Institute is looking at any preventative measures to make our patients healthier and potentially avoid cancers in the future.
Yes, absolutely. Prevention is a key mission of the Cancer Institute, and we have many investigators who are working on questions like diet, certainly smoking cessation, even though there may be no smokers in this room. It wouldn't be surprising if there's not a single one. Smoking still is a problem in our area, not as much as it is in other parts of the country. I'll just take a short segue to say that one of our missions in the Cancer Institute and as an NCI Comprehensive Cancer Center is that we have to be working to improve the health of the patients living in our catchment area, is the term from the NCI. And we determine our catchment area to be the 10 counties that ring the San Francisco Bay. That's where 80% of our patients come from for clinical care. And we do a deep dive into those counties in terms of what the risk factors are. How much smoking there is, how much alcohol use, how much inactivity, obesity, all these different risk factors for cancer. And it's not the same from county to county. There are different risk factors in different counties because there are different populations. So we are actively working on trying to mitigate those risk factors. Diet is a very important one. Again, maybe not so seemingly important in our immediate area here in Palo Alto, but definitely in our catchment area. And the obesity epidemic is a huge one in the United States and internationally as countries develop, and that's leading to severe diabetes, liver disease, and increases in cancer. And so I don't have a specific comment about intermittent fasting, but certainly diet, exercise, fighting obesity, these are all key aspects of reducing cancer incidence. And you can imagine that I showed you the good news about mortality, cancer mortality decreasing. Well, there are things that are fighting that diminished mortality, right? Which are things like obesity, which is taking over from smoking. As smoking comes down, obesity is coming up. So there's a lot of moving parts to that diminishing mortality curve. Yes.
So you mentioned at the beginning of your talk some of the disparities in cancer diagnosis and treatment, and I'm curious, as these therapies are getting more advanced, for example, with CAR T, which can be fraught with manufacturing difficulties, and then the administration is not especially simple. How do you foresee this getting into the community, and how are we going to sort of be able to deliver these complex treatments en masse?
Yes, that's a really important question. Thank you. We are actively working on these issues in terms of reaching patients in other socioeconomic groups and in terms of underrepresented minorities. We keep a close measure on this in terms of those who come to Stanford for clinical care and those who come for clinical trials. We do quite well with treating and putting patients on trial who are from Latinx populations, Asian American Pacific Islanders, for example. One area that we're actively trying to work on is in terms of our African American representation. We only have about 3% African Americans who come for clinical care, and therefore we only put about 3% of our patients on clinical trial, or 3% of those are African American. And so we're working hard to do outreach to these communities, and Stanford Healthcare and Lucille Packard are pushing the clinical care envelope outside of our immediate two counties here, San Mateo and Santa Clara, out to the East Bay. And we have clinics out there now on the adult side. In Emeryville, we have an exciting joint venture with Sutter to have a joint cancer center, and that's broken ground already in Oakland. That will be open in a few years. And through this way, we'll be able to reach some of these other populations. But I think you're even asking a more complex question, which is, for these very advanced modalities like CAR T, there are going to be insurance barriers to those therapies as well as accessing those, and it's going to require a lot of work to solve that issue. Thank you.
I wanted to know if the mission of the SCI includes children's cancer research and clinical trials. You mentioned at the beginning, you kind of dismissed pediatric cancer. That's the way you look at it from a pediatric oncologist perspective anyway. But in fact, a lot of this research, genetic and chromosomal and genetic has started in pediatric populations, yes. And the research history
sure.
is rich in pediatric.
Yes, well,
as is the clinical, trials.
Yes. So I apologize for if you felt I gave short shrift to pediatric cancer. That was not intentional. I mean, the story of chemotherapy for curing patients really comes from pediatric leukemia, right? That's what you're alluding to. That's where that story came from. Sidney Farber at Dana-Farber started those trials, So to answer your question, pediatric cancer is absolutely part of the SCI. It absolutely is. I mean, it's a smaller piece just because pediatric cancer is a smaller piece in terms of number of cases, but it's at the front of what we do because treating children with cancer is so fundamentally important. The clinical trials apparatus is part of ours at SCI, so all the pediatric trials run out of the SCI as well. We work closely with Tanya Gruber, whom we recruited as the new chief of pediatric oncology from St. Jude, and she actually is in the SCI, my associate director for pediatric cancer. So that's all fully integrated. You're right that for clinical trials, many more children are on clinical trials than adults because the history of pediatric cancer has been one based on protocols, complex protocols, and all many children are treated the same way on these protocols. So there's a much higher frequency of putting children on protocols than adults. So we're putting about 12% now of our adult patients on clinical trials. It's much higher for children, in the 50. Well, we'd like to get it to 80, but it's right now I think about 50%. Yes. Oh, yes.
Is any research being done on the proliferation of blood vessels into solid tumors?
Yes. So angiogenesis has been a very exciting area of research over the past 2 decades, and it's led to an approved drug, Avastin, which is an antibody to VEGF that mops up the VEGF cytokine that's stimulating blood vessel growth in tumors, and so it's a widely used, blockbuster drug to fight cancer. There was a lot of thought about 20 years ago that, from the work of Judah Folkman, who was a legendary pioneer in this field at Harvard, that this would be the way to cure it. And there was a famous New York Times article when there was so much buzz around this that essentially cancer is cured now because we solved the blood vessel problem. But sometimes we get a little bit too excited about these things, and we now know that cancer is very, very complicated, and it's going to be hard to cure through a single idea. But that is part of our armamentarium against cancer, and it's an exciting area.
Apparently not. Oh, there we go. Terrific. Please join me now in a round of applause for this wonderful presentation. Thank you so much, Steve. Great.